share with you today. Okay. So what I will share with you today is basically some results we've had on working on protein kinases. As you can see here, I combined two different themes: is the conformational landscape of protein kinases, and I will explain what we really mean by that, which is, in our view, provides really exciting opportunities for anyone to understand how protein kinases, but also will argue that any other dynamic proteins as well, mechanistically and functionally. But then, which probably is also a thing of importance to some of you there, is that then how someone can take advantage of this conformational landscape to therapeutically target protein kinases. And then again, also there, like I will really argue that there are a lot of advantages that currently people that don't really take advantage of, simply because there haven't really been until recently experimental approaches to facilitate this kind of approach. All right, no. So first of all, why someone would like to study protein kinases? And for those of you who are into biology, I guess this is an obvious answer, right? So you'll see here on the left all the processes that are regulated by phosphorylation, right? As you know, this is what protein kinases do. They bind to a substrate, and then an ATP, and then hydrolyze ATP, and then transfer the phosphate group to a substrate. And then this small group, okay, when it gets basically appended to a protein or another substrate, it can really have really some pronounced effects. So on the left, you see all the processes where kinases are involved, right? So you will be very hard pressed to find any process where kinases really are not really involved. And then also on the right, you can see all these different functions that are regulated by phosphorylation, meaning, of course, functions that are regulated and mediated one way or the other by protein kinases. Also, you probably know this one, kinases are some of the most common proteins mutated in disease. The number one by far is cancer, where, of course, like we have now a lot of kinase inhibitors that have been really developed for all kind of different forms of cancer. This is also similarly the case also for a number of neurodegenerative diseases and so forth. Again, like very, very important when it comes also to disease creating. So this is just to give you, I guess, an overview of what, I don't know how I can get rid of this here. All right, okay. We have a number just here, I guess, like some basic I guess statistics also about the kinases and the importance of that. We have like 518 kinases in the human genome. From that actually itself can tell you that is one of the largest and most functionally diverse, as you gathered from the previous slides. Again, from the previous slide, you appreciated that there are all kinds of different cell functions that protein kinases are involved in. And then also, as I touched upon and I said, unfortunately, the kinase activity is very often dysregulated. And that can happen by deletions, can happen by mutations, right? Can happen, for example, with fusion of genes. And I'll show you one very classical example of that causes leukemia. And that can cause, like all kind of different disease. And then I also said the cancer domain basically is one of the most commonly mutated in disease. Now, so that then is no real surprise that from a therapeutic point of view, protein kinases are major targets for a therapeutic intervention. There are currently about 80 FDA-approved kinase inhibitors, over 100 of them, maybe more, in late stages of clinical development. But something to keep also in mind is, although a very large number of these kinases, out of these 500 plus kinases, the majority of them are involved in one way or the other, in disease. Only 8% of the kinome has been therapeutically explored. So that really means that there is, there are great opportunities here for someone to go after some of these underexplored kinases. one of the major issues is in when someone develops small molecules for kinases is selectivity. And then I'll give you some insight into that, why this is really happening. And of course, if the selectivity is poor, given that you have 500 plus kinases, then also you have off-target binding, which can cause all kinds of different side effects. And something else also that it does really occur often, unfortunately, is patients that they are treated with kinase inhibitors at some point. Okay, it's likely that they develop drug resistance mutations. All right, so now let me explain first what we really mean by conformational landscape here, right? Which is, in my view, this is really the next frontier in structural biology. We are in an era where I would think, and I think that that's a common knowledge now, that someone predicting the structure of a single protein, it's a problem that has been largely solved by AlphaFold, and that's also probably the case for many complexes. So then in that era where we can predict the ground state, the energetically most stable state of any protein, right? So what is really now, what someone needs to investigate, where someone needs to go deep to really now make this connection between structure and function. What is really the missing information there? And then in my view, what is really missing is now an insight into this multitude of conformational states. Okay, so what do we really mean by that is that proteins, and that's true for not only kinases, but for any dynamic proteins, right? But in this case, I'll just show you the example, in kinases, but that can apply to GPCRs or nuclear receptors or transcription factors or like other enzymes, whatever you name this one. But the idea is that these proteins exist in multiple conformational states, right? And then each one of these states may be associated with a very different property or function. So you get, for example, one conformational state that may be related to a certain property or a certain outcome. Then you can have a different state, and then maybe again, like related to a different one. And so you have all these multiple conformational states. And typically, when someone uses crystallography or cryo-EM or even NMR for that matter, we determine the structure of the most stable conformation. And as such, all the other conformational states that in most cases can be rarely populated, maybe 1%, 2%, 5%. They have really remained experimentally, like just beyond detection, which means, of course, like they cannot be structurally characterized. And as such, all this information is really being missing. But there is now very strong evidence. We have actually, my lab and other labs, they have done now work where we have very clearly and convincingly demonstrated that having access to this multitude of conformational states, only then someone can really get a better view, closer to a full view of the function, the properties, and the regulation of these different systems. And so today, actually, I tried just to make this argument. First of all, I'll show you how someone can do this one, right? Again, I'll keep this one at a very high level so then you can only get some idea without going into, of course, details and experimental details by NMR, but how we can make this one happen. Then the kind of insight that someone can gain from getting the structures of these states. And then finally, like how someone then can exploit that, how can leverage the structures of these states now to develop selective inhibitors and ligands, and hopefully, at some point also drugs for against kinases. All right. So first of all, let me just give you an brief introduction into how a kinase domain looks like. This is, of course, the engine of any kinase. That's a kinase domain. It has two lobes. You have this N-terminal up there, and then the C lobe, which is the larger one. And then I highlight here some structural features that will be important for you so then you can really follow, first the structure, and of course, the changes that may really happen there. We have this so-called P loop here and the ATP binding site. This is under this loop. This is where, let me see if I can get the laser here, something. I guess we don't have that. Okay. Right. I guess it's been recorded. That's why. Okay. I can't really quite point to this one, but hopefully, like the slides are informative enough for you to be able to follow that. The ATP binding site. And then we have this helix that is called the alpha C helix. And that's very important because, as you can see, we have a glutamate that emanates from the alpha C helix and forms a salt bridge with this nearby lysine. That's very important because it stabilizes the orientation of ATP that is required for the chemistry to happen, right? The hydrolysis of ATP and the transfer of then the phosphate group to the substrate. We have the so-called DFG motif, which is super conserved in kinases. That's an aspartate and this phenylalanine. The aspartate is very important because it coordinates the magnesium, like together, of course, like nearby the ATP phosphate groups as well. Then the activation loop, which is another very important structural feature. And this is because at the end of this A loop, that needs to be in its extended form that forms the substrate docking site. This is where substrates, other proteins, they dock, and they point their tyrosine or serine or threonine towards the active site for then this phosphorylation to take place. Now, something which is important to keep in mind is that because of evolutionary pressure, right, kinases in their active state, they all adopt a very, very, very similar structure, right? Because there's only a certain way that you have the alpha C helix in the so-called in conformation, which is the active one, DFG in the in conformation, which also is the active one, the activation loop in this open extended conformation, so then the substrate can dock to that. So for this reason, when someone looks at the structure of any one of these 500 kinases, they all look very, very similar. And that also is a big problem when it comes, for example, to target therapeutically kinases, right? Because if the goal here is to target, okay, to design ligands that they can bind into kinase in the active state, then because they look so similar to each other, right? So then you have, of course, very, very poor selectivity. There are very good examples of that. For example, even in some cases, this is beneficial, depends on what you're really after and then how many kinases you like to target. But in most cases, that's really a problem because, of course, you target many more kinases than one you wish for. Dasatinib is, for example, one classical example that it's an approved drug, and yet it binds to like about 200 kinases instead of only the one has been designed for, which was the BCR able. Right. So anyway, going back into the structure, it's because of evolution, you have only, you have a very, very similar active state. And any departure from this structure will result to what we call an inactive state. And of course, kinases are designed to adopt both active and inactive structures, right? Because active structures, they only should really be adopted when the kinases are supposed to phosphorylate. But then they should just, inactivate themselves somehow, right? So then that really stops, because in case, and this is something that happens in disease often, then you have a dysregulated activity. So kinases, they just keep phosphorylating, and that will cause, for example, in many cases, uncontrolled growth, tumorigenesis, cancer, and other disease. All right, so let me just, here in a simple animation, show you some of these concepts that we just, discussed. This is the alpha C helix, the activation loop, the DFG motif, right? So you see now we'll have the ATP that should come when you have the active kinase, and then it will bind to the kinase. Then you have the substrate here in orange, and you can see you will have this basically hydrolysis of the phosphate, of the gamma phosphate, and the transfer over to the substrate. And then this is different ways now to make a kinase inactive. For example, this DFG motif can flip. And then if you go into the so-called out conformation, then this is not active anymore. The alpha C helix also can move away because that will break the salt bridge that is required, as I told you before. And there's another way to inactivate it, is just by this activation loop now folding back, not being anymore in this open conformation. So then the substrate cannot bind to the kinase anymore. So these are three possible ways. Of course, there are more ways, and there's also like a combination of different ways that you really may have, for example, a kinase being activated. And then, so what we've really been asking here is that, which is really very important, right? So number one is that, how many different inactive states can be there? Because as I showed you before, active states can only be one because they all have to be very, very similar. But inactive, it can be anything, right? Because for as long as you are inactive, it doesn't really matter. I mean, for as long as you can just make sure that you move away from the active conformation, right? Different kinases can adopt different inactive conformations. So then we're asking how many of these inactive conformations are present in the human kinome, right? Which is, of course, very important for us to understand the mechanisms, but also it's very important because it presents opportunities for the design of selective inhibitors, and I will explain why is that the case. Now, again, very briefly, what I said before is that when someone looks and is trying to identify, monitor, detect, and ideally also determine the structure of these very low populated states or alternate conformational states, right? How that can really happen experimentally is it's very, very challenging. And the reason is that, of course, they are very low populated, they are very close to noise. Yes, and then even when someone, for example, can detect such a state, being able to structurally characterize them and then also assign a specific function or property to them, okay, it's also like very, very challenging. So my argument here, it is that, which I think now is widely accepted, that the only really way to experimentally access these states is NMR spectroscopy, right? With NMR spectroscopy, I don't know how familiar you are with that, right? But NMR spectroscopy, of course, is one of the three tools, including crystallography and cryo-EM, that can provide high-resolution structures. But the benefit of NMR spectroscopy is that we study proteins in solution, right, at ambient temperature. So that's, of course, the closer to native environment, if you will. And then in addition to getting structures, also we can look at how the shape of the structure changes over time. That means dynamics. And then what is more relevant, like for our today discussion we have here is this detection of these low populated conformational states that actually sometimes we can detect them as low as 1% with the newest, sorry, there was a question?
No question. That's someone sneezed. Sorry.
Okay, that's okay. And then it's, as I said, as low as 1%, but also in some cases with the newest instrument and magnets and cryoprobes sometimes actually can go even down to 0.5% or something like that. And then we can determine the structure, but also as a bonus, what also we can do, which is very, very important, is we can determine the energetics, the population, that means the number, as well as the kinetics of these conformational transitions. All right, so the kinase I will talk to you about today is Abel kinase, right? And then someone is asking, now we can ask how we can really monitor the conformational landscape by using NMR. And then I use Abel for a couple of reasons. So this is the first kinase we worked on. We use this one as a model system for us to develop the approach that I will explain to you again at a very high level in the next couple of slides, and how we can access the conformational landscape. But also at the same time, because it's a major, of course, it's of very high medical importance as Abel kinase, but also because it's very well known that one of these fusion oncoproteins, BCR-ABL, right? So this is where we have basically chromosomal translocation, and then you have the BCR gene now fused to ABL kinase that will result now in dysregulation of ABL activity. BCR-ABL is the one that causes chronic myelogenous leukemia. And of course, it became famous, or rather infamous, if you will, because then Gleevec, still remaining the most famous tyrosine kinase inhibitor, and of course, one of the most famous drugs ever, they were developed about 30 years ago or so. Gleevec binds very selectively to BCR-ABL, and and this is, of course, was really highlighted also back in the day on the cover of Time, and that for a very good reason, because before Gleevec, imatinib, right, before Gleevec, the survival rates for patients suffering from CML was abysmal, like about 4 to 5%. And then with the advent of Gleevec, that actually went up more than 90%. So I don't think that there is any other drug in the history that would ever claim, like such a huge success in treating a specific disease. Right, so let me show you a little bit actually of the mechanism here, and then we'll get back actually to Gleevec, and then how now we can use what we've learned from this one to potentially develop like better inhibitors. So this is just, like we can keep this one because I know you are recording, so you can, like it's kind of like a bonus animation that we have prepared here to show this one to some of our donors, for example, and how it works with kinases, where they bind to substrate, they phosphorylate, and then what happens, how you like to, for example, target them. In this case, it's Gleevec that was designed to bind very potently, so then ATP cannot really bind anymore, right? But unfortunately, as you know this one very well, many patients develop resistance to Gleevec, and that would mean is that there's, for example, a mutation. You see here, that's the gatekeeper more often than not. So Gleevec cannot really bind anymore, and as such, ATP can bind, and then unfortunately, right, in this case, you cannot really have drugs to do this one, or at least not Gleevec, but new drugs, new generation, second generation, third generation of drugs can be designed through structure-based design to now basically bypass this mutation or fit in within this mutation and then block that. That, of course, tells you how important it is for someone to have the structure, the structural insight into that. This is how the main domain of ABLE looks like. So you have here in white, you have the kinase domain with the N lobe and the C lobe, as you can see. And then this colorful domain here is what we call the regulatory module that has an SS3 and SS2 domain that docks on the back of the kinase and keeps it in this inhibited state. All right, so how then we can, by NMR, we can access this conformational landscape, right? There's a lot of course physics here, but again, I'll keep it at the very highest level just to mention that we have one of the, largest NMR centers here with our flagship 1.1 gigahertz, the largest one actually when, and the most powerful magnet when it was installed at the time, like about 6 years ago, together with another slew of many other NMR instruments. In this case, we just label, of course, our proteins. In this case, you have a proton and a carbon-13 as topically labeled. And then as these things are really shifting from state to state, we are also trying to observe basically the movement. And then from this, we are trying to extract as much of a structural information as possible about what residue is doing what. Right, so this is typically what we do. All right, so it's because these are large systems, we cannot do this one the typical way where we just only label proton and nitrogen 15 or carbon, for example. In this case, we have to use very selective, we have to take advantage of specific isotope labeling approaches. Basically exploit how E. coli uses different metabolic pathways to label this protein specifically, proton and carbon. So here you can see all the methods, for example, all the aromatics that we label with proton and carbon-13, so then you can get the spectra and everything else being per-deuterated, because otherwise it would not really be possible to study such large systems by NMR. Okay, now very briefly also let me show you how we can do that, how we can basically detect these conformational states. This experiment is called CEST. It was first initially applied into MRI field and then has been repurposed especially from the group of Louis Cave in University of Toronto, to be applied to proteins to get the information actually that I will just explain to you now. So if you do this experiment and then in a protein that is, let's say, like for example, like a very rigid, right? There is no other conformational state. There's only one conformational state. Then you'll get one dip like this. And that's the chemical shift here that will give you very structural information. And you can do this one for every single probe, every single residue that you have in your protein. But now, if you do this one in a protein where there is additional conformational states present, right? Then you can see this dip here on the left, the pink one that shows this dip, and that is now a very strong indication that you have basically a second conformational state. Right, so that's what it did. And then when we apply this one to ABLE, as you can see, we notice that there are 3 dips. The major dip, that's the ground state. And the ground state is, for example, what someone would determine structurally if we were to apply any kind of techniques. But you can see also the other ones, excited state 1 and excited state 2, where that clearly tells us that, and then you can quantitate that that there are three conformational states, at least three conformational states, that they interconvert with each other. And then also we can measure the population, right? So the ground state is almost like 90%. And then each one of the other excited states is like about 5%. So what someone can really do this one. This is, of course, very novel information. And then the big question is, right, okay, but then what is potentially the structure of these states? So we first determine the structure of these ground states. So if I go back here, okay, so this one on the left, this is what we first determined by NMR, and this is how the structure looks like here, which is a fully active conformation. So then we know that Abel kinase, the domain itself, without the regulatory domains, just by itself adopts a fully active conformation in solution. And then we want actually, and then try to now determine structurally the structure of the other conformational states. And then in order to do this one, we cannot do it when it is like very low populated, but what we need to do is that we have to convert the ground state to excited state, so then we can make the excited state now the new ground state. And the best way to do this one is by designing specific mutations that they will shift the equilibrium from ground state and will stabilize your excited state. And then in this case, you can just go, of course, and determine the structure. And that's what we did. So then we determined the structure of each one of these three states now. And that will give you an idea here. I'm just morphing, right, animating the structural changes as Abel first transitions from this active state to this inactive state 1, right? And you can see, for example, the DFG here, this phenylalanine 401, that flips from the in state to the out state. And this aspartate 400 in this active state, as you can see now here, in this case, it can coordinate magnesium and ATP, but then in the inactive state, it flips 180 degrees. So then in this case, your kinase is inactive because it cannot coordinate this one anymore. And then there's also a second state, this inactive state 2, right? And then I also show you now how much more pronounced basically are the structural changes as Abel moves from this active state to this inactive state 2, right? And then you can see among many, many different things, I can just draw your attention when this one, as this one is looping back and forth. Look actually at phenylalanine 401 here. This is in the active conformation, and the inactive phenylalanine 401 will move all the way here to the left inside the ATP pocket. So then it blocks ATP binding, right? So this is, for example, one way also to inactivate your the kinase. The other one, if you just now, for example, look here at tyrosine 412 or 30408, this is the activation loop. They move all the way to the other side. So they close up, and as such, that cannot really now allow the substrate to bind anymore. All right, now, something actually which is very important is that imatinib, which is, of course, like Glivec, Glivec was a serendipitous discovery. There was no, of course, like the structure wasn't really known. They came up, like through some screening and, through a process. They identified Glivec, and then what's really amazing with Glivec is that it was extremely selective. It only bound BCR-ABL and then KIT kinase, and then also maybe weakly and a couple of more. I mean, maybe a little bit more, fewer, more of that, but that was it, right? So it was, in other words, actually extremely selective. And this is the reason that Glivec, even 30 years later, and even in the presence and the availability of third and fourth generation of inhibitors, still remains the first line of defense, right, for against BCR, against CML to target BCR-ABL. But then why that was so selective, it was totally unknown. And then only recently, when we worked on ABL, now we discovered for the first time what is exactly why Glivec is so selective, right? And then how it really binds to ABL. So what we found out is that when you look at the structure of Glivec bound to ABL, which is here in blue, and then the structure that we have now discovered, this 5% of this inactive state 2, they look very similar, right? And that really told us that now have identified the conformational state that Glivec selectively binds to. In other words, right, so this conformational state already pre-exists in solution, but is very low populated, and that's why no one ever saw this one before. And only recently, when we worked on actually enable, we discovered that this is, what's happening, which also explains the selectivity. Now, here actually I zoom in into the active side. You can see here now Glivec, imatinib in gray, and then you see, nevertheless, that although this kind of like fits nicely into this pocket that pre-exists in this orange state, which is the able inactive state, even there, okay, there are some very significant conformational change. For example, this tyrosine 272 in orange is all the way to the right in the inactive state. And then when Glivec binds, it pushes this one actually all the way to the left. And then what I'm trying to say here is that this is, of course, Glivec is very selective, but also it binds with a much lower affinity than other drugs, like dasatinib, for example, that only half the size. So Glivec has a very big size, and yet it binds only with about 10 nanomolar. The reason being, okay, is that still has to expend a lot of energy to rearrange the pocket. And then what I've been asking is that, all right, now we have in our hands the structure of the I2. Can we design an imatinib analog, if you will, a Glivec analog that would really fit better, so then we can now, hopefully come up with a ligand that has actually a much higher affinity than Glivec. Anyway, so that really shows you here the conformational landscape that we have now for able active, the able I1 state, the able I2 state. This is what we just described. And then how Glivec, imatinib, right? Imatinib is a generic name for Glivec, how it really binds to this selectively to this I2 state and to form the complex. Let me also, like very briefly here, tell you in a couple of slides, okay, why this approach is great to dissect the underlying mechanism of Glivec resistance mutations. All right. So one of the, as I told you before, one of the big problems that we have in patients that are treated, not only Glivec, but of course any other inhibitor, right? But because Glivec has been around for so long, we know this one very, very well. These patients develop drug resistance mutations. And some of them are nearby. For example, I draw your attention to this threonine 334-I. This is the gatekeeper. The gatekeeper now, the threonine, gets mutated to an I solution. It's very bulky. And as such, of course, because it's just next door to Glivec, it reduces the affinity, and that explains the resistance, right? But then there are also some other mutations, as you can see, that are actually very far away from Glivec. For example, this histidine 415P down here in south, right? You can see it's like so far away from Glivec. And somehow this mutation, this histidine to a proline, it's a drug resistance mutation. So how is it really possible that a mutation that is located so far away from Glivec causes this kind of resistance? Okay, let me basically explain you how is that the case and why our approach here is really so superior to anything else to explain that. Now, when someone looks at the structure, right, you only see a snapshot. You only see the final, the end structure, and that is not, of course, like the all the information you need. What you need the information for is what is the structure, the state that needs to be present for Glivec to bind to selectively, right? And as I told you, we discovered that this state is this 5% populated I2 state. So someone would hypothesize that if there is a mutation that destabilizes this state, right, then Glivec, of course, wouldn't bind anymore, or would bind with much lower affinity, simply because this state is not really present there, right? And then how we can test this hypothesis? Well, we can do this one very nicely with our approach. When you look at the red here, which is wild type able, you see the presence of the I2 state. But then when you introduce this mutation, this histidine to a proline drug resistance mutations, then you can see now that this I2 basically disappears. It's flat. And that really means now that the population of this inactive state has been reduced to less than 1%. And this is the reason this being destabilized. And this is the reason why now Glivec binds with much lower affinity. And this is the one that gives rise now to a drug-resistant mutation. Similarly, we can also use this approach to, again, from a mechanistic point of view, and also, like from a clinical point of view, in many cases, also understand the effect of the mutations on the what to call the regulatory spine, the DFG motif, the gatekeeper residue that just actually discussed before, as well as the activation loop. This is important because these are really hotspot areas for oncogenic mutations. It's very well known that many kinases, they develop this kind, they have this kind of mutations that they give rise to disease because they hijack the system and they are just messing basically with the conformational landscape. Okay, now I would like to, for the last few minutes here, I would like to talk to you a little bit about how we can leverage this approach for selective small molecule inhibitor design opportunities, right? So what is the advantage offered by our approach here? If someone targets not the active states, right, which as I told you, remember, they are very similar across the kinome, but rather target distinct inactive states, then potentially we can increase selectivity. Now, this is exactly why Gleevec is so selective. It doesn't bind to the active conformation, right? But it binds to this inactive conformation. Now, as I said, that wasn't known, right, until we figured this one out. But now we understand very well why Gleevec is so selective. And this is the question we're asking, right? Yes, Gleevec was a serendipitous discovery, and there's a reason why in 30 years, okay, there hasn't really been, like another Gleevec, right? It has been extremely difficult for anyone to come up with something, like as selective, for example, as Gleevec. But if we have now in our hands the structure of these states, not only for BCR-ABL, but for many other kinases, can then we use the structure to selectively design such ligands, right? Also, what also we've been after as well is that in these transient conformational states, very often also we have like transient pockets. These pockets are only visible only for a fraction of the time, right? They are not present in the ground state, and as such, for example, you will never be seeing in crystallography or cryo-EM for that matter, right? But if someone has a structure of this low populated states, these transient states where we can have these pockets open up, then someone also could really target those selectively. So this is, basically recaps here why this is, like so important, I guess, and then how these invisible states that we call the transient states, why they can offer new therapeutic opportunities, right? As I said before, what maybe, they are they're seen as inaccessible binding pockets. They may become accessible transiently in these states, right? We have unique structural features that are absent in this ground dominant states that we can also be taken advantage of. And the greater selectivity that I already discussed, but also the fact that in some of these cases, the ligand can stabilize specific states that modulate signaling output. Now that goes beyond the standard paradigm of targeting a kinase just to inhibit the enzymatic activity, but also it goes into selectively stabilize a specific conformational state, and then through not enzymatic activity, but through scaffolding activity, okay, which of course will then modulate the interaction, the binding partners, that can also affect the basically signaling. And then we really hope now that this approach that we are really taking here can expand the druggable space of kinases. So how do this one? We use two approaches. We use two strategies. And then the first approach is we use structure-based virtual docking. We use, in most cases, virtual flow. This is a new kind of approach that Christoph Gorgula, who was before actually at Harvard, he's now here with us, has developed, that who's a computer scientist, that we can virtually screen about 70 billion compounds for this state. So once we have then these structures, we virtually dock as many as 70 billion of these compounds, and then we order a number of those, and then we perform, of course, experimental screens. But the second approach, which is also extremely powerful, is that we don't use, in this case, computational approaches, but rather, once we have the structure of these conformational states, then we can design mutations to stabilize them. And as such, now you have these rare states, these transient states, but stabilized, so you can produce, you can express them, and you can purify, and then use these structures to screen against, like a large number, for example, either fragment libraries, DNA-encoded as well, like ligands and libraries. This is something else. This is something that now we are really working on, and then we are ready to go after to use. It's a 1.3 trillion compound library to hopefully be able to identify a number of hits. And we are, as I'll show you actually in the end, but we are working on a very large number of kinases. But for BCR-ABL, right? We use this one first as our model system, and then it's still BCR-ABL, of course it is. Although we have a large number of drugs, right? We have now imatinib, as you can see here. We have the dasatinib that I said you before, ponatinib, many GNF2 and GNF5. These are all allosteric inhibitors. There is still need to develop even more potent and more selective inhibitors, even, for example, maybe if possible, more potent, definitely, and even more selective than imatinib. So we use this one as our model system, but also because we wanted, hopefully, to develop a drug for this. And we have been, as explained to Arthur, I won't really be able to share with you more details here because this is all now under, patents, and it's all very confidential. But I'll just show you that the idea here is that what we would like to go after is that we would like to go after a certain state, that inactive state 1, for which, there are no really drugs. We know there are drugs that they bind to ABL active conformation, like the dasatinib, for example. Gleevec is the one that binds to the I2 state, but there is currently nothing on the I1 state. And because we have the structure, that's what we did. We asked, can we develop, for example, a selective inhibitor that is very, very selective for this I1 state, and as such, potentially also can be even more selective than Gleevec, and then potent at the same time? And the answer is yes, actually we can do this one, and this is not even fully optimized yet, but we do have several compounds. This is just second generation of compounds that we have now produced. And as you can see, the efficacy in BCR-ABL plus, we can have positive here, cell lines. You can inhibit BCR-ABL, and this is with IC50 of about 8 nanomolar. We have now a new analog that actually goes down to 5 nanomolar. And then we are in the process of using, basically now, of further developing and refining this strategy, with the idea to probably bring this one even further down, maybe to 1 or 2 nanomolar. That would be really fantastic. But of course, what is really in our mind much more important is to design inhibitors that are really very selective. All right, so that's it. And I just wanted to say, before actually I show all these names here, is that we have a very big consortium here, which is what we call the blue sky mechanism on kinases. So then I lead this team of about 70 people, which is, they're just listed here. They are NMR people, they are cryo-EM people, crystallographers, bioinformaticians, biochemists, ligand discovery, of course, people, cell assays people who do all kind of in vivo, in vitro cell assays, and of course, enzymatic assays, all of the different things. And we are currently studying about 150 kinases. We are doing this ligand discovery team now for a number of those. And we've had actually really great success for many of them. And hopefully, at some point also we'll really be able to, also have a drug. So I guess this is all what I wanted to share with you today. And I'm happy actually to take any questions you may have.
All right, thank you. So questions? No question? So yeah, I have a question. It has to do with the molecules themselves that are you're trying to form as inhibitors for these kinases and looking at specific states. Like, what considerations do you take for the molecular structure in terms of trying to fit them into these states, or do you just use the optimized forms of these structures?
In terms of ligand discovery, you mean?
Yeah, the ligand discovery, but yeah, in terms of what can potentially be a good inhibitor for these different states.
We take really an agnostic approach. So we have, when we do first we have the structure, and then we do this virtual docking. We go up to many billion of compounds. We screen them, we rank them. And then based on this, we have we ordered from enamine like about 1,000 compounds or so, and then we ran the usual experimental approaches, identify, I mean, it depends on what our hit rate is. Sometimes actually it's pretty good. Let's say we have like 30 or 40 or 50 good compounds, and we get crystal structures of those, and then because we need crystal structures to be able to further refine those. But then we have all kind of like different scaffolds that they can really bind to that, right? Of course, the libraries we are screening, we are using that are already in principle optimized by enamine for like drug properties, right? So for example, solubility is not really that bad, and then also the pharmacokinetics, all of that, ideally also is not really like too bad. But yeah, these are all things that you have to, of course, to refine through medicinal chemistry in the end.
Okay, thank you.
Does it make sense? Okay, yeah.
Yes, it does. Questions you guys have? All right, so if there are no questions, let's round of applause for our speaker.
Thank you.
All right. And so thank you so much and have a good day.
Thank you. You too. Thank you. Bye.